Thank you very much for the quick, short introduction. Today, I'm going to introduce our genetic breeding program approach that we performed in part of the eye efficiency project. The eye efficiency project is an ongoing H2020 project known as the Aquaculture for Innovation Action. This picture shows you the main intervention areas of the project, which the 4 main products will be developed. While the main focus of the project is to integrate the new digital solutions in the aquaculture technologies, the breeding approach developed in the presented research is a horizontal product of the project, which can support the adoption of main technology products. So what's the need behind? We know that quite well the largest cost in fisheries is the feeding cost, and especially in case of those species which require high protein content. The capture-based marine proteins and oils should be decreased in fish feeds. We know that quite well. And that would be really advantageous if we could use cheaper and alternative protein and oil sources in the feeds. For example, processed animal proteins, plant or microbial origin ones. There is an increasing usage and tests are ongoing using soybean, algae, and insect-based proteins, or plant and insect-based oil sources in different feeds. Microbes and algae would be also beneficial because they can be used for the bioconversion of agricultural or industrial waste into high-value proteins and their lipids in an industry scale. So one solution is So one solution can be if you're using these alternative protein sources, and we know that from previous studies, a large amount of soy meal in the diet could decrease the growth of farmed animals and fish species. Although from rainbow trout studies, we know that the selection-based studies showed that the survival rate or even the mean body weight and biomass can be improved after even just one selection, a single generation period. So it seems that the genetic selection is a suitable way to select and adopt the population utilizing a plant-based diet. African catfish as a species is a great model to test these new feeds and the genetic selection-based breeding program. So in our project, we performed a growth rate and feed utilization broodstock selection program through 3-generation crossings and selection, where African catfish were used in half industrial scale and in a flow-through system. In the studies, we used commercial control feeds and a low fish meal experimental feed and compared the effect. Size selection was performed repeatedly in the different generations, as you can see in this diagram. The target, the main goal of this study was to create an African catfish line, selected line, which similar or better performs in growth rate compared to those lines which are fed with conventional feeds. It could reduce the production cost and provide more sustainable farming methods. And finally, we would like to isolate genetic markers for the prediction of phenotypic traits and marker-assisted selection in the near future. Where we are now and what are the current results? Comparing the body mass of the groups in the F1 generation, we could see some difference related to the sex in the utilization of the feeds. Although this difference was not significant between the groups, but it seems that the experimental feed was better utilized in case of females in the F1 generation. For the body mass distribution in the F1 generation, we could conclude that the groups fed with the low fish meal diet had a lower growth rate compared to the control group in the F1 generations. When the F2 generations were compared, the selection had a remarkable effect on the growth rate. The selected lines grew significantly faster compared to the non-selected groups fed with both the control or even with the experimental feeds. The calculated average gain was around 14% in the F2 generation. We have to note that these studies are still ongoing, so further comparison is needed. We still have more than 1.5 years in the project. But the current conclusions that we already can gain is that the genetic selection had a positive effect on the growth rates. It seems that the utilization of the feed is affected by the sex of the fish, although the third generation, the F3, should be investigated and genetic marker identification is in progress. We can conclude already that the selected F2 lines better utilize both the conventional and the alternative protein source feed, which makes the new line commercially more attractive. And we hope that during the analysis, of the third generation, we could generate a subline which will utilize the alternative protein sources much better than the previous ones in F2 generation. So thank you for your attention. And if you have any question related to this topic, then we are happy to answer.